Waiting for your signal, Judith.
We're now live on Facebook, Tracy.
Lovely. Thank you very much, Judith. So welcome to all of our friends and colleagues in the CUH family to another presentation brought to you today by the Reach Network as part of our Black History Month celebrations. I'm delighted to welcome Adel Helmy, who'll be presenting today on neuroinflammation and brain injury, as part of a series of presentations we've been bringing on wellness and health. Black History Month is also an opportunity for us really to showcase the wonderful diversity and contributions that the BME workforce are making to Addenbrooke's. And we've already had some fantastic presentations this week from people like Dami Collier, and Adele with us today is certainly no exception of that. I'll do a very brief introduction before I hand Adele the platform, but I'm quite proud of Adele because he really is, I think, true homegrown talent. And what I understand is that Adele actually completed his undergraduate and higher neurosurgical training here at Addenbrooke's. Adele, in addition to his wonderful portfolio of work, has also edited the Oxford textbook of neurological surgery and has national roles within NICE, contributing to the UK guidelines on the management of head injury. Adele also chairs the National Neurosurgical Audit Programme here in the UK, so making some absolutely fantastic contributions in his area. And so I can hand you the floor now, Adele. We're all really looking forward to hearing what you've got to say and showcasing your work. Thank you very much for joining us today, Adele.
Thanks very much, Tracy. That's a really kind introduction, and I'm very grateful to have the opportunity to speak to you all today. So it's a fantastic scheme of work that you've put together, and yeah, delighted to be part of it. So I'll just share my slides with you and just speak perhaps for the next 45 minutes or so, just about some of the work that we're doing within the Department of Neurosurgery. So I have dual roles. I'm an honorary consultant in Addenbrooke's. I'm a neurosurgeon, so I do operations on calls and so on. I work clinically, but I'm also an associate professor of neurosurgery within the academic department. And I do research largely into traumatic brain injury, but also in other types of brain injury like subarachnoid hemorrhage, for example. And really this scheme of work, and I'll talk about various things, is really a real team effort. So although I'm talking about it and been involved in many of this work, you know, this research really spans a huge number of people within the department, a large number of collaborators, and really for all clinical research, it's a team game. So lots and lots of other people who should be credited with this work. So just to give you an idea, we talk about TBI, traumatic brain injury. And the reason we call it that rather than head injury is when people bang their head and they break their nose or they get a cut on their forehead, that's unfortunate and unpleasant. But as neurosurgeons, we're not interested in that. What we're interested in is brain injury. And that's why we prefer this term TBI. And I'll give you a little bit of background about what TBI research has been like over perhaps the past 20 or 30 years, and then a little bit about the work that we've done more recently. I'll talk about neuroinflammation. This idea of inflammation in the brain is something which is relatively new, as in it came about in the past 15 or 20 years. Previously, people thought that inflammation didn't happen in the brain because it was immunoprivileged, but we'll come on to that. I'll talk a bit about neuroprotection, so drugs that might protect the brain after it's injured, and then a little bit about future directions at the end. So the way that head injury has been managed, perhaps for the past 25 years, is this idea of secondary injury. And what that means is that when you have a head injury, that injures the brain or it injures the neurons. And after that, all sorts of other things can happen that can make things worse. And we call that secondary brain injury. And that can be low blood pressure, hypotension. It can be low oxygen, hypoxia. And this piece of work, which is quite old now, came out of something called the Traumatic Coma Data Bank in the early 90s out of the US intensive care system. And it showed that if you had a single episode of hypotension, so blood pressure lower than 90 millimeters of mercury, or you had a single episode of hypoxia with oxygenation below, it says 60 millimeters of mercury here, but that's 8 kilopascals in our language, then that doubles your chances of dying. So we showed that it wasn't just the head injury that caused the problem, but it was the effects after the head injury and the consequences of that on the brain that could also worsen outcome. And that really guided the management of head injury in the ensuing time. And it led to this concept, and there's this little sort of vicious circle here where you get the primary insult, brain injury, that can lead to brain swelling, that can cause raised pressure within the brain, that then reduces the blood flow in the brain, that reduces oxygen in the brain can't generate energy, and that makes more brain swelling. And so the cycle turns, and that makes things worse and worse for people. So really the thinking was, what we need to do is we need to find a whole load of monitors, and I've given some examples here, an ICP sensor, intracranial pressure monitor sensor, to measure the pressure in the head, measures of blood flow in the brain, measures of oxygen in the brain. And if we can figure out how to measure these things and make them better, then we can reduce that secondary injury. And that's been a very, very successful scheme of research that's been going on over many decades. And it's really led to the establishment of specialized neurocritical care. And you'll know in Adam Brooks Hospital, we don't have one critical care unit. We actually have lots of critical care units, one of which is the neuroscience critical care unit. And certainly when that was set up in the early 2000s, that was the largest facility in Europe by size for neurointensive care. And what came in was all of these different monitors in order to look at the brain, identify all these secondary insults, and try and jump in and treat them before things got worse. What we found was, with this increased monitoring, we found that things got a lot better. And I've got this stylized cartoon of this sort of plateau effect that you see as you introduce more and more monitors. And this is actually from the business literature. But if you think about adding these additional monitors, adding these additional interventions early on, when we made sure that patients had enough oxygen and had enough blood pressure, that was making major impacts. I talked about the doubling of mortality when you have these very severe events. But then as time goes on, the ability of those monitors to add more and more benefit becomes harder and harder because the easy gains have been made. And we end up with this problem of diminishing returns, where the incremental benefit of adding on yet another monitor, yet another minor gain doesn't lead to changes in our patient care. And what that means is that we need to start thinking in a completely different way about this problem, not just thinking about these secondary injuries, but thinking about what is actually driving this primary injury in the first place, and how can we address that in order to improve patient outcome. So this is really what brought us to inflammation. I don't know if anyone recognizes this gentleman. This is someone called Celsus. It actually says it on there. And in Roman times, the historians credited him with defining inflammation. And inflammation in those days was rubor, calor, dolor, tumor, and functio laesi. Many of us don't speak Latin anymore, but what that was meaning was swelling, redness, pain, and loss of function. And that's how we used to think of inflammation, probably for 2000 years, all the way since then. And in some ways, until relatively recently, that's how we thought about inflammation as well. But there's a slightly more refined and scientifically specific way of looking at it. And the way that I'd like to look at it as the endogenous response to injury. So inflammation happens everywhere. Anytime you have an injury, that's not just to the brain, then you get this body's response to it. And that response to injury may be to any insult whatsoever. I mean, we're going to talk about trauma, but it can be towards infection, it can be towards autoimmunity, it can be in any particular context. And when we think about it that way, inflammation becomes considerably more complicated than the way that Celsus originally envisaged it. We can think about inflammatory cells. That's what I mean by cellular. We can think about inflammatory mediators, things that are produced by those cells to communicate. That's what we talk about with humoral. We can think about acute inflammation, things that happen quickly, chronic inflammation, things which are still happening weeks or months later. We can think about inflammation in different parts of the body, not just in the brain, but are there inflammatory mediators in the blood? Are there inflammatory mediators other places around the body that could impact on that? And we can also think about what we call innate inflammation, which happens the same way every time, and adaptive inflammation or immunity, where the parts of the immune system can generate new responses, typically through antibodies. And if you've heard Dami speaking recently, and obviously the COVID pandemic, it's made us all experts in immunity, immunization, and so on. So there's considerable complexity to inflammation, but the message that I like to bring across is that it's any of these endogenous responses, and those responses have obviously evolved or developed to our benefit, but in some circumstances it can cause a lot of damage. We talked about brain swelling, for example, being a damaging initiator after head injury, and by understanding this inflammation, the hope is that we can intervene and try and make things better. And this is a cartoon from one of our papers that tries to illustrate many of these points, and you can see that there's all sorts of different players. They're in different parts of the body, they're developing over time, they're interacting, and our work has been trying to sort of delineate this better with the idea of ultimately therapeutic intent. So the way that we started looking at this is to look at these mediators. How do these cells communicate and how do they drive inflammation? We look at these two types of molecules, cytokines and chemokines. So cytokines are these molecules which are made by cells which drive these different inflammatory responses, and chemokines are ways that we can draw in new cells to initiate these responses. And we can look in lots of different compartments. So we can take blood tests, that's what I'm talking about with plasma. We can look at cerebrospinal fluid, that's the fluid that's around the brain. We can measure cytokines and chemokines there. And we can use this particular technique called microdialysis, which looks at the brain extracellular fluid. And I'll speak specifically about microdialysis because that's a lot of the work that we've done here locally in Cambridge and that I've been involved with myself. So I hope this isn't too gruesome for anyone, but this is how we manage people on intensive care. This is a patient with an intracranial pressure monitor inserted, and we do it through this bolt, which actually allows 3 probes to go in. And one of them is a pressure monitor to measure the pressure in the brain. One is to look at brain tissue oxygen. You may remember that was one of the secondary injurious mechanisms that we talked about. And the third is microdialysis. And you can see this is a scan of a patient's head, and you can see the 3 monitors sitting there. And these monitors actually sit within the brain substance. And here, this is what a microdialysis catheter looks like. It's got a tube that goes in where we pump in fluid. It's got the semi-permeable membrane where substances within the brain can diffuse into that fluid, and then we collect the fluid again in this microvial, and then we can analyze it for whatever we like. And this technology really grew up looking at markers of metabolism, lactate, pyruvate, glucose, and so on, as another marker for the type of secondary brain injury processes that occur. But we've used it in more recent times to look at these inflammatory mediators as well from a research perspective. And this is a scanning electron micrograph of a monitor that was put in someone's brain. You can see this is what the semi-permeable membrane looks like under very high magnification. It's a bit like a sponge. And here you can see actually some cells have migrated into it. So this is something that was actually taken out of someone's brain. This is a cartoon of what the microdialysis catheter looks like at the microscopic level. So we pump this fluid down. It allows exchange of these mediators between the brain and the inside of the catheter, and then we collect this fluid up again. And we've done all sorts of basic science work on the bench, looking at how these molecules get across, how big they are, how much charge they have, and so on, to allow us to recover these mediators, these cytokines and chemokines. And this is early work that we published showing a whole range of different mediators. These are the names of them here: interleukin-1 alpha, beta, IP10, which is a chemokine, and interleukin-2. And what we were able to show is we can measure these things in the brain using this microdialysis technique. These 2 different lines are differences in the fluid composition that we used in the microdialysis. There are methodological issues that we developed to try and look at that. And we also looked in blood as well and see how the 2 compare. And what that showed us was that there are specific responses for each of these cytokines and chemokines within the brain. It's not that they all respond in the same way, and we can detect that using this particular technology. But particularly, we don't see the same responses necessarily in blood. So this is brain specific, it's being driven by the brain, and that gave us a confidence that this may be an important marker of the sort of injurious mechanisms that happen after head injury. This is a little panel which is showing a comparison of the concentrations between the brain and the plasma in the same patient at the same time. This is from human brain injury patients here at Addenbrooke's. And we can see for some cytokines and chemokines, the concentration is 60 or 70 times higher in brain than it is in blood. And what that suggests is that this is coming from brain and leaking into the blood rather than the other way around. Some people are saying, well, it's just because they've broken their leg or they've broken their pelvis. That's why that inflammation is being detected in the brain. It's a passive leak, but the concentrations are several times higher in brain than they are in blood. We then wanted to look and see, is there a specific pattern to this response? And we found that we were able to detect a peak for the cytokines and chemokines in lots of different patients at different times. And by amalgamating these peaks from different patients, we were able to show that there were some cytokines that came about very early, some in the middle and some late. And really that corroborates a lot of what we understand from the animal literature. So people can go and do these experiments in mice or rats or other animals, and we can show that those responses are recapitulated in man. We can actually detect them dynamically within our own patients. And that's giving us confidence that, again, this is contributing to the secondary injury mechanism. We can use more sophisticated statistical techniques to look at that rather than just looking at the peak concentration. This is a technique called principal component analysis, which I won't bore you with, but essentially it shows us that there is this consistent change over time and this consistent pattern across all patients. It's a highly conserved response. So I don't know if anyone recognizes this panel, this historical picture. I don't know if people can speak up. Probably they can't if we're live streaming, but this is Caesar crossing the Rubicon. So this is in the 1st century AD. So Caesar defied the Roman Senate and crossed the Rubicon, which was a river, with his army, therefore calling into question the authority of the Roman Empire of the day. And I don't know if this is true or not, but he's quoted as saying, alia iacta est. I'm not going to do this entire talk in Latin, but alia iacta est means, and so the die is cast. And what he was getting at is that by crossing the Rubicon, he had set in chain an inevitable sequence of events that led to war in his case. And we still use that aphorism, crossing the Rubicon. And it's a similar thing with inflammation after head injury. What we are finding in these patients is once they have the head injury, alia iacta est, that cytokine and chemokine response inevitably follows. So we see this as a consistent pattern afterwards. And this really changes our understanding of what we thought of as primary and secondary injury, because people thought primary injury was something that just happened in the instant that you had the head injury. But in fact, after you have the head injury, there is this irretrievable consequential sequence of events that happens that we see with this cytokine response. One of the things we've been able to do, this is work that we did with some collaborators in the Karolinska Institute in Sweden. We were very interested in this idea of the crosstalk between inflammation in the periphery, that means in the blood, from other types of injury, and inflammation in the brain. The reason that's important is that many of our patients, when they come into intensive care, they don't just have brain injuries. These are people who've been in unfortunate accidents, road traffic accidents, or perhaps assault or something of that sort. And they have lots of other injuries as well. And that might be injuries to their bones, or it might be injury to their internal organs. And obviously, while they're on intensive care, there's all sorts of other things can happen. So, for example, ventilator-associated pneumonia. These patients are very sick in a coma, and they're intubated and being ventilated, and they can develop chest infections, for example. So does that affect the inflammation in the brain? So I won't labor this point, but we were able to look at a very large group of cytokines and chemokines in blood and brain, and we were able to look at what are called partial correlation coefficients, how they relate to each other between blood and brain. And we were able to demonstrate that actually there is bidirectional crosstalk between the brain compartment and the blood compartment and systemic inflammation. So inflammation is being driven by the brain in these patients, but what's happening in the rest of the body also has an impact too. So the brain inflammation can change the way that the blood inflammation occurs, and inflammation in other parts of the body, either from other injuries or from infection, whatever else, can also affect the brain. So as a conceptual tool, it's important to realize that this doesn't happen in isolation. And of course, from a therapeutic perspective, it makes things considerably more complicated for us, both in terms of interpreting this sort of data, but also in terms of thinking about therapeutic angles. How can we try and make these patients better? One of the things that we were interested in when we got this data was thinking about, what are these cytokines doing? Are they doing anything harmful? How do they relate to each other? Does one cause another? So release of one cytokine acts on a cell and leads to another effect. What we did was we took some of the information and data that we had from the human patients, and we went back to the laboratory with collaborations with various groups within the university. And we used some of the technology we have in the university. These are primary cell cultures. So these are cultures of human cells in a dish. And what we can do is we can take the cytokines and chemokines we know are in the brain, and we know the concentration they're at, and then we can treat the cells in the dish and see what happens to them. And that can help to tell us which of the cells that are in that dish are responding to the cytokines, and therefore try and infer whether it's the cells that we know that are in the brain that are responding in the same way. And we've done that in various different model systems. We've done it with neurons, we've done it with astrocytes, and more recently we've done it with microglia. Again, I won't labor the point in terms of the exact responses, but the point I'll make is that by using this sort of data that we get from the humans, it helps to design the preclinical studies, the studies that we're doing back in the laboratory. So previously we were finding that some of these studies were using concentrations of cytokine that might be 1,000 or 10,000 times larger than we're seeing in humans. So if that's the case, then the sort of results that are produced by those studies are not informative or representative of what we're doing in the human. So we sometimes call that reverse translation. We do a study in humans. The data that we get from the humans helps to inform the model system that we use. And we've done all sorts of work. These are some pictures of, these are astrocytes, in fact, and neurons here and astrocytes here within the brain. And we treat them with these cytokines and we see the effect it has on them. We were able to use some sophisticated techniques to look at the receptors that are on these different cell types. And that helps to predict which cytokines and chemokines they can respond to. We've done something similar with microglia. This is a paper that we're currently putting together now and hopefully will get published later in the year, showing again the same principle. And microglia, which are the macrophages of the brain, if you like, are the cells that tend to respond most to these stimuli. So we think probably the microglia are the most important cell type in our particular context. So this is a picture of the Holy Grail, and this was a concept in Arthurian legends. That there was this object that would have this special powers to confirm, amongst other things, protection or healing. And in traumatic brain injury, our holy grail is that of neuroprotection. Can we find a drug or intervention that protects the brain after injury? I've talked a lot about traumatic brain injury because that's the model or the pathology that I work in. But this concept can be extended to all forms of brain injury, stroke, hemorrhage on the brain, some forms of neurodegeneration. So really the key thing for us is, can we find a drug that helps? And the problem that we have is that all the previous trials at phase 3 for neuroprotective therapies in traumatic brain injury have failed. That's not from lack of trying. There have been dozens of studies out there. And in simple terms, you can say that everything works in rodents. The reason these trials were carried out is because people did studies in animals and they found that they were of benefit. And then when we try them in humans, they've all failed. And there's a very famous quote which says, insanity is doing the same thing over and over again and expecting different results. So if we keep trying these studies and they don't work, should we still be doing them? Does anyone know who that is attributed to?
Einstein?
It is. So that's Albert Einstein, a very, very famous quote from Albert Einstein. So the question that we asked ourselves is, what are the reasons that these studies aren't working? And is there a way that we can use our methodology, in particular microdialysis, to assess that? And part of the problem that we have when we do something in a rodent and then we try and do it in a human is that things are very different in the human situation. So, for example, when we talk about timing of drug delivery, in a lot of the rodent studies, they give the rat the drug before they give them the head injury. It's very difficult to imagine for a human, you get up in the morning and you get in your car and unfortunately have an accident, how we could ever give you that drug before you had the head injury. We can't predict when that happens. It's like Minority Report. There's an issue about whether the drug gets to the brain in sufficient concentration. Does it get across the blood-brain barrier? There's this important structure around the brain which protects the brain from what's going on in the periphery and stops important mediators and also drugs from getting into the brain. So in these rats, they're sometimes injecting the drug directly into the brain. So when we then give that drug to the patient intravenously or by mouth, it never gets into the brain. So perhaps that's why it's not working. The next question is, are we getting enough of it into the brain? How do we know what dose to use in the human? So we can think about the dose per kilogram in rats, and there are tables that you can use that help to translate that into what might be useful in humans. But how do we know it's getting into the brain at sufficient concentrations to carry out the biological effect? And then finally, and this is a really important problem for us within neuroscience as a whole, where rodent models are used very, very commonly, is, does the rat brain look anything like the human brain? Is there a fundamental difference in the biology of rat and human brain? And if you ask the average man on the street, they'd probably say, yeah, there is a huge difference. Obviously, our brains are very well developed for things like thinking and planning and, all the things, language, all the things that we think of as making us humans. Rats' brains are very good for navigating around a space and for smell, because that's what rats do by and large. In the neuroscience field, we talk about gyrencephalic and lysencephalic species. Lysencephaly means smooth brain. A rat's brain doesn't have lots and lots of convolutions. It's relatively smooth, while a human brain is gyrencephalic, which means it has all of these grooves and dips, which you see on any cartoon that you might see of the brain. And the advantage of that from a biological perspective, it gives you a lot more brain squeezed into a lot tinier area. And we think that the lysencephalic species and the gyrencephalic species may have fundamental differences in the way they're put together. And therefore, that might be a reason why things are working in these model systems and they're not working in man. There's got to be a reason why. So what we said was, well, can we use this technique, microdialysis, which I described to you earlier, to help to address some of these issues? So one of the issues is pharmacokinetics. Pharmacokinetics is a posh word for saying, does the drug get into the right place at the right time, at the right concentration? And the second thing that we can look at with microdialysis is biology. Is the drug doing what we expect it to do based on what we've learned from animal literature or some of those cell culture models that I showed you earlier? And what we tried to do was use a microdialysis methodology to try and address these questions. So in terms of which drug we chose, I mean, I've been talking about inflammation for the past 25 minutes or so. You won't be surprised to hear that we've tried to use inflammation as a therapeutic target. So if we know that these mediators are there, we think they're doing something important in the brain, and importantly, we think that they may be responsible for a mechanistic role. They're not just there as bystanders. They're driving the important changes that happen after head injury, like brain swelling, like the cells being unable to generate enough energy. The other advantage of inflammation, as I said to you earlier, is the therapeutic window. The time in which we can intervene may be extended. So like I was saying earlier, there's this sequence of events that happens after a head injury. There's not just at the instant that the person injures their brain when actually we don't have access to the patient. We can't be giving them these drugs. These events and these processes are happening for many, many days afterwards, and that gives us an opportunity to intervene. That's what medically we call a therapeutic window. And the third great advantage is that actually in lots of other fields, largely in diseases like the rheumatological diseases, there's a whole host of drugs which have already been developed which address inflammation. And that allows us to do what we call repurposing, which means we take a drug which has been used in one context and we know it's safe. And the drug studies have already got demonstrated that it's got a good safety profile and it's cheap. And then we try and address it in our population of patients, in this case, traumatic brain injury. And the molecule that we chose in this particular study is something called interleukin-1 receptor antagonist. Interleukin-1 is the prototypic inflammatory cytokine. It's a cytokine which is really important for driving inflammation, not just because it's called interleukin-1, but it does drive inflammation. Actually, interleukin-2 was the first interleukin detected, but it wasn't recognized as an interleukin at the time, and that's a slight aside. When we look at interleukin-1 receptor antagonism, which means blocking the receptor, it's been found in a whole range of animal models to be effective. I've described some of the limitations of the animal models, but it's still important to demonstrate that it can be beneficial. And it's a drug which was developed again for rheumatoid arthritis as a third or fourth line agent. It's a drug that's in the BNF, and you can find it. It's called anakinra. And using some sophisticated genetics, it's a recombinant drug, which means that you get a vat of E. coli. We think of E. coli as bad, but you get a vat of E. coli and you present it with some mRNA, and then the E. coli themselves can make this protein for you, and you can purify it and give it to humans. And luckily, the drug company's done all that for us, so we don't have to bother with that. And in particular, in humans, some of the observational data that we had was we looked at the balance between two cytokines, interleukin-1 beta, which is the thing that's acting on the receptor to potentiate signaling, and we looked at the antagonist. The antagonist is also made by the body, and the two sort of work in a balance. And we find that with a lot of systems in biology where you have very potent mediators, the body actually makes its own antagonists. There's this intrinsic check and balance that occurs. And we compared the outcomes of patients who had high interleukin-1 beta to R1 ratio with those who had low ratio. And you can look at these patients here and these patients here. And the ones in this group, these black dots, are the ones who had a good outcome by dichotomized GOS. I won't get into too much into that, but they had a good outcome. And these patients had a bad outcome, these ones in the squares. So again, that's adding rationale to the fact that maybe trying to change this balance between interleukin-1B and interleukin-1 receptor antagonists may be of benefit. So we set up an open label randomized controlled trial. So we gave 10 patients in intensive care the drug and 10 didn't get the drug. We looked at patients who had a GCS 8 or less. That means people with a bad brain injury who are in coma requiring ventilation intensive care. We tried to focus it on patients who have diffuse brain injury. These weren't patients who were coming in with a blood clot which needed removal, because we thought that that was a slightly different patient population that might benefit from this drug. And we gave this drug 100 milligrams subcutaneously every day for 5 days. We looked for adverse events. Does it make the patients unwell or cause complications? And then we used the same sort of methodology that I described earlier. We measured a whole load of cytokines and chemokines in the brain to see if the drug is getting into the brain. We measured the downstream effects, and we also looked at the effects in blood. And what we were able to show, this is a panel showing the concentrations in blood. The red is the patients who were given the drug. The blue bars are the ones who weren't. And you can see initially, before we gave the drug, the levels are commensurate. And then when we give the drug, surprise, surprise, the plasma levels go up. And that was in keeping with what we knew from the pharmacokinetics in plasma for the drug in the rheumatoid arthritis literature. So that was telling us that we were giving the drug as one would expect. But more interestingly, we were able to look for the drug in the brain as well. So these are patients who have these microdialysis catheters in the brain, and we were able to measure the concentration in the brain. And you can see here, again, the red bars are the patients who got the drug. There's this gradual accumulation of the drug the more doses we give. So you see the concentration going up, starts to dip down. We give the drug again, the concentration goes up, and so on. Now, you'd think that this would be an obvious thing to do, but actually this is the first description of measuring drugs in this way to demonstrate that they actually get into the brain in the target concentration we expect. And then what we were able to do is not just measure how the drug got into the brain, but also a whole range of cytokines and chemokines, and see, does giving the drug have an effect? So this is, again, a principal component analysis plot. These blue dots are data from the patients who didn't get the drug. The red dots are data from patients who did get the drug. I won't elaborate too much on how we get these principal components, but you can think of it as an amalgam of all the cytokine and chemokine data that we've got. And it's showing that the drug is having a biological effect. The drug is having an effect on these cytokines and chemokines in the way that one would expect. And we were able to show in a more refined way that if we split the patients into the group of patients who had the highest concentrations in brain and the patients who had the lowest concentration in brain, what we've called greater and least responders, that correlates with the change in the cytokines downstream. So that's suggesting that it's the drug that's causing these effects. And what that allows us to do is superimpose. I showed you a cartoon like this of the cellular responses after head injury right at the beginning. Superimpose the effects of interleukin-1 receptor antagonism on these different cell subtypes. So that's telling us something much more refined about the interaction within the brain, but it's also telling us that this is potentially something which we can build on in terms of designing future studies. And what we'd really like to do next is look at a dose escalation study. What we were finding is that some patients were responding, some weren't, and that would depend on how much drug we could get into the brain. And the obvious thing on the back of that is to say, well, maybe in those patients or in some patients, we need to be giving more drug. Now, if we hadn't circumvented all of this and just gone to a phase 3 study with that dose, of course, we wouldn't have had that information. And I think this is really the great advantage of using this sort of approach in terms of designing clinical studies to help bridge that gap, what we call a translational gap, taking those promising preclinical findings and actually delivering them in human patients. Sorry, these slides are for some reason duplicated. I will just flick on from those. So that sort of paradigm, that sort of approach to carrying out drug studies we've continued with. And this is a study which is ongoing today. This is a study which we're currently recruiting to. It's called Pangea. It's actually a commercially funded study. It's looking at another molecule of interest, something called substance P. There's a lot of evidence out there that substance P is important in blood-brain barrier breakdown and causing brain swelling. And we're using a very similar methodology to the one that I described before, where we're giving this drug in patients who've had a bad brain injury, obviously as part of an ethically approved study. And then we measure the pressure within the brain. We see how much drugs and interventions they're getting to control the pressure in the brain. But we're also looking at things like scans, MRI scans, to see whether we can reduce the edema, the water that's on the brain. And we're also looking at the cytokines and chemokines in exactly the way that I've described for the previous study to see, can we detect a change when we deliver this drug? And we've done this study in two ways. We've done a dose escalation study in exactly the way that I described earlier. Can we get the right concentration in brain first? And then we're going to follow that on with a double-blind placebo-controlled trial. Can we show that there's a difference between the patients who are given placebo or the patients who are given our drug? So we see this sort of approach really as a platform, a way of carrying out these studies in future that addresses different aspects of inflammation in order to try and make these studies more fruitful, rather than doing the same thing again and again and expecting a different result every time. So those are my slides. I hope they weren't sort of too complex. I don't know who's dialing in terms of the audience, but I'll be delighted to take any questions or comments. And as I say, thanks again for giving me the opportunity to speak to you today. Thank you.
Wow, Adel, amazing, absolutely mind-blowing. Before I even begin to comment on the wonderful things that you said, I just want to, for anyone who's dialed in or watching the presentation live on Staff Facebook, please feel free to use the chat and put any questions in there for Adel, and we'll navigate them so that Adel answers. But I'm absolutely stunned, actually, for so many reasons. I'm just going to try and articulate, Adel, some of my thoughts, I think. So first of all, this is just a wonderful example of the CUH research strategy for the BRC in terms of bench to bedside. You know, this is just the perfect example. And I just think what you're doing is just amazing. I mean, it's been many, many, many years since I've thought about interleukins and, you know, all of that language. And I'm like racking way back into my memory thinking, oh my God, this is really sensational. And I think what you're doing is amazing. I just wanted to actually get your thoughts on something, actually, that Alea Iacta Est, I can't remember if I said that correctly, crossing the Rubicon. Actually, I'm really interested in crossing that Rubicon with you. You know, this correlation between the blood and brain crosstalk. I just wondered, Adel, if there was any correlation, or if there was any thinking in amongst yourselves, your peers, that if there's any relevance for this to other illnesses like neurodegenerative diseases or autoimmune diseases, because I feel like the inflammation piece is huge and this could go, you know, in so many different areas. So I just wondered what your views were on that, and for you.
Yeah, so I think you're absolutely right, Tracy. Thanks very much for the kind words. I'm mildly embarrassed, but thanks for the kind words. But I think this idea of inflammation not just being in one compartment has really exploded. There's a lot of interest in gut microbiome. I don't know if you've come across that idea that actually the bacteria in your gut can have all sorts of different effects on you. There's a lot of interest in the relationship between that and some neuropsychiatric disorders, for example. There's a lot of interest in, the type of food that you eat, whether it changes your gut bacteria. That can affect not just your mood, but your metabolism as well. There's a lot of work that's been done around Parkinson's, in fact, looking at, again, changes within the gut that can affect that. The difficulty we have, because it's a relatively new concept, no one would think that your gut would affect your brain necessarily. One of the really interesting concepts that we're trying to flesh out is quite how that happens. Is it that there's something to do with immunity? Because the gut, in fact, is the greatest, we think of our skin as the outside world where we interact with our environment, but because the gut is very long and tortuous, actually, in terms of the surface area, the gut is actually a huge surface area where we interact with our external environment. And obviously it's not a clean environment for the majority of it. So there's loads and loads of bacteria and potential immune interactions there. And whether that's generating an immune response, which is then traveling to the brain, whether there's soluble mediators, there's things coming out of those bacteria which dissolve in the blood, which then go to the brain and then can have an effect. And for example, in the neuropsychiatric literature, could it be something like serotonin? There's a whole range of drugs that target serotonin, for example, that can affect mood. So quite what that relationship is, honestly, nobody knows. But I think you're absolutely right. This idea that the brain was in an ivory tower, there was this idea that the brain was immunologically privileged because of the blood-brain barrier. So inflammation just didn't happen in the brain. All of that now is being overturned, and people are sort of thinking in very different ways about the interaction between these body systems and looking for the causes of these conditions in different body systems as well. So it's certainly an exciting field, but it makes it a little bit more complicated because we're all working slightly outside our comfort zone, outside the organ which we all grew up learning about in our various trainings. So yeah, absolutely an area of active and fruitful research.
So exciting. I'm excited just listening to you. I think you're a pioneer. I think it's absolutely amazing. And I'd be really interested just to keep in touch, actually, to see how things develop, particularly if you decide to explore anything around the gut, because I'm a celiac, and sometimes even if I'm following explicitly a gluten-free diet, I find that my stomach, for whatever reason, my gut is just angry and unhappy, and I can almost feel stuff going on in my body through my own intuition, but not really understanding what's driving that or how that's working. So I think, I think that's definitely an untapped resource. Just amazing. The microdialysis stuff, again, I think that's just an area where, I mean, there's just so much where that could go in terms of obviously I know this is your area, TBI, but do you imagine, or can you foresee diversifying that into any other areas or any other collaborations that you're thinking about exploring?
So, I mean, I think that's an excellent question. And there's two ways that it's sort of been taken forward. One's from a clinical perspective. The great advantage we have, and it was a technique that was really established by Pete Hutchinson, who's our professor of neurosurgery. He really brought it into the unit as a clinical tool, first and foremost. And the great advantage is when we have these clinical tools established, all of that research can follow. When you talk about the BRC and the sort of successive translation, a lot of that is predicated on having, a successful and dynamic hospital where we're treating patients and we're using this for clinical benefits. And that's, the research follows from that. So I think from the clinical perspective, the other condition that we'd really like to establish it in is subarachnoid hemorrhage, which is a type of bleed on the brain that happens from an aneurysm. And that's one of my other clinical interests. And I think there, from the clinical perspective, certainly. In other fields, obviously in the neurosurgical conditions like those conditions, because we're operating on these patients, either to put in monitoring or operations to remove blood clots or treat them in other ways, there's the opportunity to insert those monitors. And those patients, when they're treated on intensive care, there's the infrastructure and the resource and the fantastic nursing staff who are able to gather all that data and store it and use it. Outside that, I mean, we have put microdialysis catheters into patients who've had brain tumors. Sometimes you can use it in stroke patients. There are lots and lots of other ways that we can use that technology, but that's largely been driven by research, because the practicality is we wouldn't normally put those catheters in unless we've got that specific research question. But you're right, it's a tool. I think that's the way to look at it. And if you've got a question like, does a drug get into the brain? How can we address that? How do we find out? Then, for that specific question, it's an excellent tool to be able to address that.
Yes, it's amazing. And I just wondered, actually, where you were speaking about aneurysm and these sorts of patients, and obviously this is a Black History Month, part of our celebrations. I just wondered, from a clinical perspective, have you noticed or are you aware of any trends in demographics that might manifest in as much as the Black and Asian communities, for example, tend to, like with sickle cell or diabetes, more prevalent in those communities? Have you noticed, or do you suspect, or would you be interested in exploring any sorts of trends in demographics?
Yeah, so I mean, I think that, it's a fantastic question. I mean, just starting with subarachnoid hemorrhage, I mean, it's a disease that's often associated with high blood pressure and smoking, and high blood pressure can be associated with certain demographic groups, certainly. I mean, with traumatic brain injury, with TBI, sadly, it's a disease which is often associated with poverty or deprivation. So we see a higher incidence, and that can be for all sorts of different reasons, either for risk-taking behavior or drugs and alcohol or violence. And one of the things that we're really interested in, particularly with our PPI, our public and patient engagement, I'll be very interested if there's anyone on this call who'd be interested in that sort of work, is reaching out to a greater diversity of communities. So part of the problem is when we do PPI in Cambridge and sort of in some parts of East Anglia, it's a very homogenous population ethnically, and it's a very well-off population. We're very lucky to, live in and around, Cambridge is an affluent area, but we actually cover a much larger population of patients within East Anglia because we're the regional neurosurgical centre. So there are some areas like around the coast, for example, there's been a lot of work from government, in fact, about deprivation in coastal communities. And there's a lot of coastal communities in East Anglia who aren't as affluent and have a whole range of different problems, and they may be susceptible to TBI, but also they may not have the necessary sort of societal or sort of personal support in recovery after a head injury. And we also cover areas, further south, perhaps around Luton or Bedford, which are much more ethnically diverse and a very, very different population from that which we've seen in Cambridge. So as a regional unit, I think it's important that we think sort of beyond our immediate boundaries and, beyond, what we see in front of us. And, from a personal perspective, I'd be very interested in engaging the whole region. Often when you invite someone to something in Cambridge, it's the people who live in and around us who can attend. But the virtual interaction is sort of become part of life. Like this meeting, we'll talk, it's not going away, and there are many advantages, some disadvantages, but many advantages. And I think engaging with a wider spectrum of people, both ethnically diverse and from areas of potential deprivation, is really, really important.
I think you hit the nail on the head there, because actually we're obliged to. There are a multitude of current NHS initiatives, including bringing the research where the research is needed. So the coastal communities, like you've mentioned in Fenland and Wisbech and Peterborough, all of those areas have lots of pockets of hidden deprivation. So actually designing lots of wonderful fancy protocols that are actually not what is needed and not where the people that need the research is happening is one of our NHS goals. And then there are lots of other initiatives like the Core 20+5, which is the 20% of the population that is struggling the most. And then regionally, our plus areas are the areas like these areas of hidden deprivation and disenfranchised communities like the Gypsy Roma Traveller or Black and Asian or Bengali. We've got all of these, like you say, we're wealthy and white for the most part in the affluent parts of Cambridge, but then as a region, actually, there are huge pockets of deprivation where this stuff just isn't happening, and it needs to happen. So I'd be really keen to perhaps pick up with you outside of this forum, sort of just plans and strategy around that. And I know that Miles Parks and the BRC is thinking about that as well. That's actually written into our bid, into our pledge, into our commitment. So this is something that we've committed to doing. So that would be really, really good. To think about.
I'll be very grateful, because I was involved with some of the PPI around the BRC, addressing, exactly those points. I mean, we're absolutely on the same page. And I'd say, if anyone else is listening, if this is live streamed, genuinely, we want to hear from other people, because obviously we dream up these applications based on, my understanding or understanding. And, we genuinely want to make a difference and help people. But, one of the things with public and patient involvement is we need to hear from the public and patients. Maybe they have a different view, or maybe they have new ideas, or maybe they've sort of something that we haven't. So, the widest engagement possible would be great. Thank you.
Wonderful, Adel. That's great. We've got a few minutes before we close off, but as this is a Black History Month presentation, you're a BME member of staff, you're a leader in your field, you're doing sensational work. I just wondered if you would be prepared to share a few pearls of wisdom from sort of your journey in terms of what's encouraged you and kept you going, to reach the academic excellence that you have. And also, if there's any staff member watching this later, perhaps on catch up, who might be interested in the work you're doing, what sort of advice you could give them of how to get involved?
So, I mean, I think, obviously the disparities that we see, whether it's around race or whether it's around deprivation, that's something else that we've mentioned, are out there. And I don't think we should try and make light of them. I mean, obviously we want to be optimistic but I think those barriers are there, and some of them are very subtle barriers. People talk about imposter syndrome, or they feel that there's a degree of tokenism. There's all these issues which I won't labor now, but I think in my own career, or in my own sort of personal development, I think there's lots and lots of opportunities out there, and I think people will come across situations which make them feel uncomfortable, or they feel perhaps they're not treated fairly, and who knows if it's genuine or not, or if there's any bad intent, but these situations arise. But I think we're very lucky within the university and within the hospital that there are lots of opportunities out there, and that's the way that I'd look at things. So you need to look for where those opportunities are, and just run with them. And you never know, some things work out, and it's like that in research. You go down one path. I haven't told you about the 100 things that didn't work. I can assure you there are 100 things that did not work, but those aren't the things that necessarily you need to dwell on, but you don't know in advance. So I think you need to go into it with that sort of positive mindset. I think as a society, as institutions, that things are changing. My personal view is that to change people's views and beliefs is a generational thing. It's very difficult to take people who've grown up in a particular environment, and perhaps it's unfair on them to expect them to change. They've grown up with a set of beliefs or views, or a particular outlook. It's sometimes very difficult to change that in a short period of time. But I think as a society, more and more awareness of issues around discrimination or inequality, and that's an important step in terms of changing that. I'd be happy to support anyone if they're interested in neurosurgery, interested in research, they want to contact me. I'll be delighted to, perhaps you could pass on my email, or you can find it on the internet. I'll be delighted to speak to anyone or link up with anyone. Similarly, anything around PPI, or even if it's some general advice. I think one thing that we can all do is this idea of mentorship. So if people do have a problem, if they've got something that they need help with, any of us, I'm sure, would be delighted to help. And it's not to make light of the tough times, but that there are opportunities out there, and you can find your way.
That's amazing. What a lovely thing for you to say. And I think finding the positives is what I was hearing out of your message. And I think you're absolutely right. There are a lot of opportunities in Addenbrooke's, and we are fortunate, but we are, however, on a journey. And as you said, not to make light of things, there is still quite a lot of work to do. But I'm really grateful to you, Adel, for sharing, a snippet into your story, into your background, into the work that you're doing. I think it's absolutely amazing. Judith is, again, yes, couldn't agree more. An absolutely fascinating presentation. I think you're one of our royal diadems, in the Addenbrooke's crown. I think what you're doing is going to make fantastic contributions. You are already, not just now, I think for the future. And I would be really keen as well to just reinforce what you've just said there in terms of mentorship, and perhaps we could again take that offline and have a conversation about maybe doing some work with the Reach Network. And how we could maybe support some members of staff who are interested in this work, give them some guidance around that. And I'd love for you to come back another time, Adel, and share your developments, in a year or so, see where you are and how you're doing. So
that's really kind.
No, you've been really, really kind. Amazing. Thank you, Adel, for your time. Thank you for your presentation. And if any questions or anything come along the way, I'll be sure to filter them your way. And if you wouldn't mind sharing your slides so that we can distribute them to anybody who would be interested. Is that okay?
So I think just some aspects of the slides, anything, because there was a couple of pictures, with patients and things, we'll have to take those out.
Yeah, redacted.
And I think all the data otherwise is published, so we can share it. There may be one or two slides which have unpublished data. Again, we're not allowed to disseminate that before it goes into publication. But I can certainly, curate these slides, and then we could share them.
Perfect. That would be wonderful. Thank you very, very much. Lovely to see you.
Pleasure. Thanks very much to you all, and thanks for your kind attention.
Thank you. Take care.
Take care. Bye. Bye.